Okay, so we're going to talk about eukaryotic cells and how they compartmentalize or look at the organelles of a eukaryotic cell. Remember, prokaryotic cell versus eukaryotic cell. Prokaryotic cell, what doesn't it have? A membrane-bound nucleus. So prokaryotic cell does not have a nucleus. Anybody else know what a prokaryotic cell doesn't have? I'm seeing if you remember from
Divide.
It doesn't have organelles, so no nucleus and no organelles is huge for prokaryotic cells. So let's look at the difference here. Maybe stay closer to the computer. My batteries are fine last hour. All right, All right, so the basic structural and functional unit of every organism is one of two different types of cells, prokaryotic or eukaryotic cells. No. Only organisms of what we call the domains bacteria and archaea consists of prokaryotic cells. So let me draw this up on the board here. So what we've done is taken all living things and compared and contrast them in their characteristics and divided them up into three different groups. These three different groups we call domains. So you have the domain bacteria, and the domain archaea, and then the domain what we call eukarya. This name comes from the types of cells that they're made of. These guys are made out of eukaryotic cells. These two right here are prokaryotic cells. And so we, humans, are in this domain. And so we're made of many cells, but all of our cells are eukaryotic. These guys here, prokaryotic cells, these guys are unicellular. All of them are unicellular. Una means one, so they're one cell big. And often, any type of bacteria are clumped in this category. So it's kind of confusing because this name, domain bacteria and domain archaea, there are some types of bacteria in here. They just classify the bacteria, the unicellular prokaryotic cells differently. They found some major differences between these two. They used to, and this kind of shows you how science changes over time and how we classify organisms as something that has changed over time. They used to clump both of these together and said all bacteria, they used to call it all bacteria together into one category. And then as more research has been done, they found that, hey, there's some differences of this type of bacteria and this type of bacteria, enough that we're going to put it into two different categories. And so that's what they did. And so to relate this to maybe what you've heard of before, is then the domain eukarya, this can be then subdivided into kingdoms. And so, you guys have heard of kingdoms, right? Okay, so the animal kingdom, the plant kingdom, the fungus, all right, and so on. And so there are certain kingdoms within here, certain kingdoms within here, and certain kingdoms within here. So it used to be that there was no domains at all, and there were just 6 kingdoms. Then before that, there were 2 kingdoms. They put everything alive and clumped it into the kingdom, animal kingdom and the plant kingdom at one point. Then they realized, whoa, there's a lot of differences between what we're putting in plants. And so then they changed it and put it into 6 kingdoms. And now we go, oh, there's more, there's these kingdoms and these organisms have some things in common, so we're going to clump them into a domain. So things kind of keep on changing as new information comes available. So one day probably this will be different, all right, and as we learn new things. So these guys are prokaryotic cells, these 2 domains. Then the protists are unicellular eukaryotic cells. So like an amoeba, paramecium, you guys heard of these before? They are clumped in this protist category, fungi, animals, and plants. These guys would be the different kinds of kingdoms here, all right? And they're all eukaryotic cells. So let's then look at what is the major differences. Then we've already kind of looked at that, prokaryotic versus eukaryotic cells, what makes them different. So comparing them. So basic features of all cells. So this would be in prokaryotic and eukaryotic cells. So all cells have a plasma membrane. Plasma membrane is the same way, you can say cell membrane as well. So cell membrane and plasma membrane can be used interchangeably with one another. There's a semi-fluid substance called the cytosol. You've probably learned it as what? Cytoplasm. Cytoplasm, right? And I just want to, we don't need to know the technical difficulties for the scope of this class, but your book has both the words cytosol and cytoplasm in it. I just want to talk a minute about that. The cytosol is the technical term for the actual semi-fluid substance in the cell. So it's filled up with this fluid substance that all the other organelles can be floating around in. And that's technically the cytosol. The cytoplasm is defined as the space between the nucleus and the plasma membrane is the cytoplasm. And so eukaryotic cells that have a nucleus have the cytoplasm, and then this is the cytosol. But for this class, you can use them interchangeably. I'm not going to nitpick about cytosol versus cytoplasm. That's beyond what we need to know. But I just want to address that so you're not confused if you look in your note in the book and it talks about different both of those. All right, and then another thing that all cells have is chromosomes. And chromosomes are made up of DNA, and remember, DNA has genes in them that contain instructions to make our polypeptides. And then they have ribosomes. So basically, the bare minimum that the cell needs to stay alive is it has to be able to make its own proteins. And so you have DNA containing the instructions to make your proteins, and you have this spot, the ribosomes, where we make the proteins. Plasma membrane just separates the outside of the cell from inside, and all of this is taking place within that cytosol, that everything's floating around it. That's the bare minimum for a cell, the smallest unit of life. So when we talk about prokaryotic cells then have all of those things that we just wrote because it says that all cells have them. But another way that prokaryotic cells are defined is by what they don't have. And so prokaryotic cells do not have a nucleus. They do not have the nucleus, sorry, I thought we were going to do the next one. I got mixed up here. They do have DNA, but it's not bound inside of that nucleus. So when we look at a prokaryotic cell, they have DNA, but it's in a specific area inside the cell. It's not bound by a membrane, and they call that the nucleoid region where that DNA is found. They do not have membrane-bound organelles, so including the nucleus, so no mitochondria, no endoplasmic reticulum, Golgi apparatus, any of those. They do have the cytosol, bound by the plasma membrane. So the cytosol, that semi-fluid substance, so they have the DNA, and they also have the ribosomes floating around in the cytosol with the DNA in that particular region. And this gives you a generic kind of picture of a bacteria, which is a type of prokaryotic cell. So if we look here, this is a computer-generated picture. And this is an electron transmission electron microscope picture of it. So let's look at the computer-generated picture. Here you have this purple, this is the bacterial chromosome. The chromosome is made out of DNA. If I were to actually take this out, if I could physically take the DNA out of the bacteria and unravel it, it'd be a big circle. So bacteria have one big circular chromosome, and the circle would be way bigger than the actual bacteria cell. So there's this huge circle, and then it all compacts into this nice little nucleoid region inside. So then it has the cytosol, the fluid inside, the little dots here are the ribosomes. You have a cell membrane, plasma membrane surrounding it. Right outside of there, you have a cell wall. What other type of cell do we know has a cell wall? Plant cell. Plant cell, right? And so plant cells and bacteria both have cell walls. They're constructed differently. They're made out of different molecules. What was the molecule that plant cell walls are made out of? Cellulose, okay? So these guys have a different construction. And then on the outside they have sometimes it's like a sticky substance, it's called a capsule. And we'll talk more about that when we look at bacteria a little bit more in depth. But to aid in movement, they have flagella. And so they can have 1, 2, this one has 3 flagella to aid in movement. All right, so that's your typical prokaryotic cell of bacteria. Eukaryotic cells, on the flip side, are characterized by having additional things. So what do they have additional then? They have DNA in a nucleus, and that nucleus is bound by a membrane called the nuclear envelope. They have membrane-bound organelles. So example, mitochondria, Golgi apparatus, endoplasmic reticulum, lysosome. They have cytoplasm in the region between the plasma membrane and the nucleus. So like I said, that region between here, so that's cytoplasm. And eukaryotic cells are generally much larger than prokaryotic cells. So when I drew on Friday, like what you would see with a microscope, you can see in a eukaryotic cell, like an animal cell, you can see inside of it, you can see the nucleus as a darker object, and then what you would see looking at bacteria, remember, are just small little, almost like dots. You can see them and see that something's there, but you can't see inside of it or anything like that. With our light microscopes, using an electron microscope for that. So let's talk about the plasma membrane, something that's in common with both. The plasma membrane is a selective barrier that allows passage of things like oxygen, nutrients, and waste. So it's to service the volume of every cell. So you have to have certain things go into the cell, things that the cell needs, and get rid of waste. What would be an example of a waste of a cell?
Carbon.
Carbon dioxide, absolutely. It would be an example of a waste. And so we have to, the plasma membrane is selective and it allows certain things in and out, so it does allow oxygen and nutrients in and waste to get out. And this is the construction of the plasma membrane. Next chapter, chapter 7, is all about the plasma membrane, so we'll go in a little bit more depth with it then. But the plasma membrane, if you don't look at the pictures, the main constituent or component of the plasma membrane is a bilayer. What molecule makes up that bilayer? Phospholipids. Phospholipids, right? And so the phospholipids, that's what we have here. We see the hydrophilic heads and the hydrophobic tails in this bilayer. Also embedded within that bilayer are some proteins. Some proteins, which are these purple guys, go through the whole lipid bilayer. Some are just on the surface, all right, the interior surface. Let's talk about the ones that go through the lipid bilayer. Can you see, I'm not sure if you can see on yours, do you see how there's this one here almost looks like it's cut in half? It has this hollow cylinder in it. Do you see that? All right. So why do you suppose that there's this hollow cylinder in the middle of this protein? What's the purpose of this protein? To let stuff in. So that's right, Nathan. So like, what happens is that The hollow cylinder and the protein has a specific shape and it allows specific things into or specific things out of the cell. And so when I say specific, that means that, for instance, one of the things that we said that going back here that needs to get into the cell is oxygen and nutrients like glucose, for instance. So you have certain proteins with certain channels that allow glucose in. Those are different proteins than proteins that allow water into the cell. And so the proteins are very, very specific. So in order to get things into and out of the cell, you have to have the right kind of proteins to allow those things into and out of the cell. And so, so that's one of the ways that it is selective, that membrane is selective, is by what kind of proteins and what kinds of things it allows into and out of the cell. All right, then the last part of the cell membrane here is these things that says carbohydrate side chains. So notice here you have these little rings hooked together. Each ring would be a what? A sugar, a monosaccharide. And so these sugars are hooked together. I'm going to use an analogy with the job of the carbohydrate side chains. It's kind of like ID tags. So it's one way that one cell can tell the difference between that type of cell and another type of cell, is these carbohydrate side chains. So certain cells, different organisms will have different carbohydrate side chains. What makes them different? The type of monosaccharide they're made of, how long they are, their branching, and so on. So kind of the shape of them. And so it's one way that your body can, in cells of an organism, can say, oh, this is a cell that's part of us, or this is a cell that is foreign to us, and so we might want to destroy it. So like, for instance, in our immune system attacks, like if we get a bacterial infection, your immune system will fight against that. How does it know that the bacteria is not part of you? All right, and so how it knows is because, in part, of these carbohydrate side chains. Looking at this is an actual picture of it's an electron micrograph picture of the cell membrane. So what would, you see these two lines, what would actually the two lines be? What part of the phospholipid? The hydrophilic heads. The black part here would be the two lines of the hydrophilic heads, and you see the white space in between? That's where the hydrophobic tails would be, all right, in between there. So let's talk about the size of cells. We all know that cells are small, and I want to talk a little bit about why. Metabolic requirements set upper limits on the size of cells. So metabolic requirements, what does this mean? Metabolic or metabolism is like chemical reactions that occur inside your cell. You need certain things for those to occur. A requirement is something that is needed, right? You need it for, in this case, the survival of the cell. So some of those things that your cell needs, those requirements, would be like oxygen, water, nutrients like sugar, all right, and so on. We need to get those things into the cell. And then on the flip side, we need to get waste products out of the cell. So Joe would give us an example of CO2 as a waste product. So we have to get waste out. And so it says the metabolic requirements set limits on the size of cells, meaning that all the stuff we need or need to get out of the cell limits the size of the cell. We don't have large cells. So let me ask you this about why it's important, or what's the benefit for multicellular organisms like us? We're made out of billions of small cells. What's the advantage of that versus being made out of 50 larger cells? What's an advantage of being made out of, made of many small cells? May I have an idea?
I guess like if you have a lot of them, if one like fails or dies, then you're not dead. But if you have like a bunch of big ones, if one fails, you like die.
Yeah, it could be like 10% of your your body. Absolutely. Did you guys hear what he said?
Right. So like, because since there's so much
cells in their bodies that I don't know, it's like a bigger variety for I don't know how.
What do you mean by different things?
Like, I don't know, DNA. Like, why? It's all the same DNA in our body, but I don't know how to word it.
Okay. I don't want to put words in your mouth. I think maybe I might know what you're talking about. Like, are you talking about like different, when you say variety, are you talking about variety of different types of cells?
Yeah.
Okay, all right. So variety, we can have variety of different types of cells that have specific jobs or functions. Am I putting words in your mouth?
Well, yeah, but that's like what I meant.
Okay, okay. All right, so we can have nerve cells and cells of your immune system and cells of your reproductive system and skin cells, and all of those have different jobs. They look different, they and behave different. So absolutely, all right. So we can have specialized functions. Anybody else?
More complex functions.
Okay, and can have more complex functions the greater variety we have. So absolutely, absolutely. So we're more complex than some other still multicellular organisms like a clam. All right, all right, absolutely. So let's look at this next bullet. It says the surface area to volume ratio of a cell is critical. So let's talk about what this ratio is. So first of all, we need to make sure everybody understands what we're talking about when we talk about surface area and what we're talking about when we talk about volume. So I have my fake Lysol cell here. All right, so this is a cell. Can somebody tell me in layman's terms how would you describe the surface area of this? Don't tell me how to calculate it, I don't want a number. I just want in just normal everyday language, how you would describe the surface area of my large Lysol cell.
That would actually be
It is a cylinder. I don't want the math. I want you just to tell me. Shut the door. When you are describing a surface area, how would you tell my third grader who doesn't know what surface area is, what the surface area of this is?
It's just the size of the outer surface that can be touched and is open to external.
Yeah, the external environment, right?
Yeah.
All right, and there are mathematical equations that we could figure out that surface area, that part of the cell in this case are this container that is exposed to the environment, all right? And so you would figure out the surface area of a cylinder different than you would a cube and so on. So that's your surface area. How would you describe in layman's terms what volume is? What's the volume of this? Joe?
How much can fit inside the cylinder?
That's right, so the space inside. All right, so then we figure out what's called a surface area to volume ratio of cells. And so we get a ratio by dividing these, and I want to talk about why this is critical. Let me use an analogy of ice and melting ice. If we took 2 ice cubes, or cubes of ice, one small, one large block of ice. So let's say we literally put an ice cube over here and had a huge, big cube of ice over here. You should be able to tell me which one's going to melt first, right? Which one would melt first? The smaller one. Can somebody tell me why the smaller one, though, would melt first? What's the logic behind that, or what's the reasoning why that smaller one would melt first? Shane?
Because it has less surface area, therefore, like, the heat's gonna melt. Like, it's just gonna, the melting time is the same, but since it's less surface area, there's less things to melt. So,
Is there less? There's overall less surface area, but less compared to that, if we actually calculated the surface area, you're right, that this would be a larger surface area than this one, but is the volume the same?
It's just less of something to melt rather than melting something as big as that. Because I mean, having a cube this big, we've got only a certain volume.
Mm-hmm.
Like a small volume model with a huge cube, we've got a lot of volume. And I mean, if it's at room temperature, it'll melt at the same rate, but the small cube will melt faster. it'll melt at the same rate, but it'll be done first, because there's less of it.
Okay, yes, and so then the volume, when we look at the smaller one, with the smaller one, the amount of surface area is a greater percentage compared to the volume, all right? So there's a greater volume of this whole thing actually touching the surface. Does that make sense?
Yeah.
Versus here, while it has a larger, if we actually look at the surface area is larger, but because it has this big volume inside of here, you have all this ice here that's not being exposed to the surface. All right, so when we compare the surface area to the volume, it takes a lot longer for that heat to get to the center of the larger block. So what physically could I do to that larger block to speed this up?
Cut it into smaller pieces.
Break it into smaller pieces. If I break it into smaller pieces, I'm not changing the volume, but what am I changing?
Surface area.
The surface area. I'm making, so the volume stays the same, but now we have a bunch more surface area, and it's going to speed up that melting and make it quicker. And so that's the same idea with cells. With cells, we need to have certain things get in and certain things out. And you can't have a cell that's so big, because what happens is that the surface area compared to the volume goes down. So while water and oxygen and sugar could get into a large cell, it wouldn't be very efficient. Those things would not be able to diffuse and get to even the center of the cell fast enough to keep up the rate of chemical reactions that they are needed for and to keep the cell alive. Same thing with waste. The waste, if the cell is too big, the waste would eventually diffuse and get to the edge of the cell and diffuse out, but probably not before they accumulate and become toxic for the cell. So therefore, we need to have cells that are small and have this, and look at this surface area to volume ratio. So as a cell increases in size, its volume grows proportionally more than its surface area. So proportionate is the key word. So this is a greater volume, and the surface area doesn't grow as to match that. So this picture here, it shows you an illustration of this. So what we have here are these cubes. These cubes are supposed to represent cells. And so, and then each, in a cube, each side is the same length. And so this says here number 1, that means each side is 1, and they're using just arbitrary units. We could use inches or whatever. So 1 unit. And so this one's 5, and then this is made out of many smaller cells that are 1. So what they're doing here is comparing the surface area, volume, and getting a ratio. So it tells you in parentheses here how to figure out the surface area of a cube. So the sum of the surface areas, height times width of all box sides, and times the number of boxes. And so the surface area of one side is 1 times 1. It says, but it's the sum of all box sides. How many sides are on a box? 6, right? So 1 times 1 is the surface area of one side times 6 sides, and then we only have 1 box, and so that gives times 1, and that gives us a surface area of 6. Our volume is height times width times length, so 1 times 1 times 1 gives you a volume of 1. Surface area to volume ratio, take the surface area divided by the volume, and so 6 divided by 1, you get 6. Notice here we do the same thing with the larger cube. So our surface area increases, so because it's 5 units, all right, the length, the width, and the height are 5 units. So we have a greater surface area, 150 versus 6, and we also have a greater volume, all right, there's a greater space, the length times height times width, 5 times 5 times 5, the volume is 125. We divide this out though, and so if you look at the amount of surface area compared to the volume, 150 divided by 125, we only get a ratio of 1.2. This tells us, like I was just going this over here, if the cell gets too big, that the efficiency of things being able to move into and out and be able to spread throughout the volume of that cell goes down. And then the purpose of this last column is to show you that this and this, they have the exact same volume. The only thing different is the surface area. So they're made out of 125 smaller cells versus 1 larger cell making up the same volume. So the volume is the same between here, but the surface area, because there's many small cells, goes up dramatically. So when we do the surface area to volume ratio, 750 divided by 125, we get a high surface area to volume ratio. What does that mean? The amount of surface area compared to the space is very great. And so why is that? Imagine this is a multicellular organism like water. Water can get in between the cells and go to the cells in the middle and be absorbed even in the center. And it doesn't have to be just on the outside of it, all right? And so what do we want? A high or low surface area to volume ratio?
Higher.
High, right? So think about mathematically, surface area on the top number, volume on the bottom number. We want way more surface area compared to the volume, all right? And so the higher the better, and it becomes more efficient. So that is another reason why we're made out of billions of small cells. So, so let's look at these small cells. We're going to take a look at our eukaryotic cells. So eukaryotic cells have internal membranes that partition the cell into organelles, and each organelle has a special function. Internal membranes facilitate cellular processes by minimizing competing interactions and by increasing surface area where reactions can occur. So let me give you an example here. Here's a cell. First of all, the internal membranes. I want to talk a minute about their construction. We talked about cell membranes being a lipid bilayer. We have the mitochondria as one of our organelles inside of our cell. If we look at the outer membrane of the mitochondria, it is also a lipid bilayer. So it is made out of membranes very similar to the cell membrane. So this would go around here, this lipid bilayer. The mitochondria also has a second layer. It has an inner membrane that folds. What's the purpose of that inner membrane being folded? What do you think? So this is the inside. This is where cellular respiration takes place. That's the process that occurs inside the mitochondria. The outer membrane that's smooth, inner membrane that's folded.
Is it to break up cells? I mean, not cells, sorry. To break up proteins? And
not really to break up proteins. There are proteins embedded in this. This is also a lipid bilayer. You guys have an idea? The curvature. Yeah?
It gives greater surface area?
Yeah, the purpose of this is that this increases surface area. So without knowing anything about the mitochondria, if I look at this, I would say, okay, the inner membrane has an increased surface area. That means that inner membrane, it must be beneficial in some way because they want more of it, all right? It's not just smooth, all right? You have, it curves and so on, and so the inner membrane actually is very, very important in the mitochondria for cellular respiration and making energy for the cell. And so, so that's what it means by increasing surface area where reactions can occur. So whenever you see folding like that of membranes, you can be sure that that membrane is important in some way for whatever processes is occurring there, all right? And membranes and membrane-bound organelles in eukaryotic cells localize or compartmentalize these intracellular, inside the cell, metabolic processes, chemical reactions, and specific enzymatic reactions. So therefore, all the things, all the enzymes and all the things that are needed for cellular respiration is in the mitochondria. It's not in the nucleus, all right? So everything that's needed for the nucleus is in there. The rough ER, all the things that are needed for there is in that organelle, and so on and so forth. So we have compartmentalization for efficiency. So every membrane, says the basic fabric of biological membranes, this is every membrane within the cell, is a double layer of phospholipids and other lipids. So that's what I was talking about here. So the cell membrane, the plasma membrane, the membrane of the mitochondria, the membrane of the nucleus, this phospholipid bilayer, they all have similar construction. And plant and animal cells have most of the same organelles. They just have some that are different. Can somebody raise their hand and tell me What's an organelle that plants have that plant cells have that animal cells do not? And
Chloroplast?
chloroplast is one. So plant cells have chloroplast. It's another one, Nathan. There is a vacuole. There's a large, what we call central vacuole. We're going to go over this, so if you don't remember it, that's fine. What other major difference you should know? Plant cells have cell walls made out of cellulose, right? Absolutely. So a lot of the organelles are the same, some are different. This picture here shows you a generic animal cell. I say generic because do all animal cells look like exactly like this? Absolutely not. So they have different shapes and sizes depending upon their function. So you can see a lot of these probably sound familiar. So the nucleus, the rough endoplasmic reticulum, and the smooth endoplasmic reticulum. You can see here the mitochondria, you can see the smooth outer membrane and the folded inner membrane. It's hard to see the ribosomes, but there are ribosomes, Golgi apparatus, and so on. One other thing I want to point out is some animal cells have a flagellum to aid in movement. Not all animal cells have that. The only animal cell in humans that have a flagellum to aid in movement is a sperm cell. And so not all cells have that. The cytoskeleton is the framework to give the cell shape. And it says microfilaments, intermediate filaments, and microtubules. Micro means small, filament means kind of like a thread. And so they're thread-like versus microtubules are like small tubes. And they go all throughout the cell and help to, they're kind of like the scaffolding system inside the cell to give it shape. So what we're going to be doing is we're going to take a look at each one of these organelles and just go over and review what they do and how they work together and so on. The animal cell, here's a generic picture of an animal cell, and you can see some of the differences here. So we have the chloroplast, you guys mentioned. Here's the large central vacuole. So the central vacuole stores water and other components we'll talk about. And then on the outside is the cell wall that animal cells do not. We'll learn also that there are passageways that connect one plant cell to another. So you can see these kind of holes between the plants. It's called the plasmodesmata. And so things can move. It's one way of how cells can communicate with one another is things, molecules moving into and out of the cell. All right, so that's your basic plant and animal cell. This just hits home the idea that they're all plant and animal cells kind of are shaped a little bit differently. This is an example of a human cell from a uterus, lining the uterus. This right here is a free surface. This will be inside the uterus. So whenever you have an organ, like a stomach or anything like that, you have space inside, and one side of the cell is facing that space, and then you have cells lining that organ, and that's what this is showing you here for a uterus. So this is open space. It's connected to connective tissue here, and this outlines one cell. It's hard to see where one cell ends and one cell begins. And you can see here, you have the folding here, increasing surface area and so on. This is a yeast cell. Yeast is a fungus, and yeasts are unicellular. They're budding. That's one way that they reproduce, is that they bud off of one another. This is a scanning electron microscope. It scans the surface, so it's more 3D. This is a transmission one that shows you inside of the yeast cell. And then here's a plant cells from a duckweed. And I just wanted to point out, you see how the human uterus cell was like a column? So is the plant cell. So therefore, we can't really classify animal cells as always being round, all right, and plant cells being rectangular, because you can have some that are not. And so that hits home with that. And so this, you can see the chloroplast, the green here, in there, and so on. This is a unicellular green algae. It's one cell big. It has, you can see, a little flagella. So it's green algae there, all right? And that's the cross section. Yes?
So the shape and function go together. Do the uterus cell and the duckweed cell have any functions in common?
No, they do not. That's a good question, but they do not. The uterus has those also, like certain areas in our body have cilia as well, to the little structures here to aid in movement and so on, but no, those two do not. Alright, so what I'd like to do next is I have two short little animations of a plant cell and an animal cell of what the basic functions of some of the organelles are and what they do. Hopefully this is a little bit of a review, but the purpose is I want to show this to you to have it a visual in your head so that when we go and talk about these organelles you can like think about it, okay? So they're not very long, but they're little animations here. Maddie, will you turn off the light? Yeah. I have to do it from the website here.
Even if you're just studying, a lot of work is going on in your cells. Let's zoom down and check it out.
It's kind of disgusting. Let's zoom in.
We've arrived in the space between two cells. A sticky coat called the extracellular matrix holds the cells together. Each cell is surrounded by a flexible plasma membrane with an incredible number of projections, docking stations, and channels. Let's dive into one of these channels to enter a cell. Whoa, look at this place. These girders and cables make up the cytoskeleton, the structural framework of the cell. They also serve as tracks for transporting cargo from one place to another. All this activity in the cell requires energy in the form of ATP molecules, which are made here in the mitochondrion. Notice the outer membrane and the inner membrane with its numerous infoldings. Many of the molecules involved in making ATP are built into the inner membrane. All those folds increase the inner surface area, enabling more ATP to be made. Moving towards the nucleus, we pass by layers of internal membranes. The nucleus is enclosed by a double membrane called the nuclear envelope. Let's enter the nucleus through a pore. The nucleus houses the genetic material of the cell, DNA, which carries the blueprints for making the cell's proteins. Almost 2 meters of DNA is crammed inside the nucleus. How does it all fit? The DNA is wrapped around proteins like thread wrapped around spools. Look, this section of the DNA has unwound and a different protein has attached to the DNA. DNA is being used as a template to make mRNA. mRNA molecules travel from the nucleus to the cytoplasm, carrying the instructions for making specific proteins. In the cytoplasm, a ribosome clamps onto a strand of mRNA. The ribosome ratchets along the mRNA, building a new protein. Some proteins stay in the cytoplasm. Others, like this one, are processed in special compartments within the cell. Protein processing and certain other metabolic activities occur in the endomembrane system, the cell's network of internal membranes. The endoplasmic reticulum, or ER, is part of the endomembrane system. There are 2 types of ER, rough and smooth. Rough ER is covered with ribosomes. Smooth ER lacks ribosomes. Lipids are made in the smooth ER. Let's go inside the rough ER. Note that you can still see the ribosome on the outside surface. The ribosome is manufacturing a new protein, which continues to grow inside the ER. Completed proteins move to the edge of the rough ER and depart in a vesicle that buds off from the ER membrane. Some vesicles fuse with the Golgi apparatus, another component of the endomembrane system. In the Golgi, proteins undergo further processing. Finished proteins are then packaged in vesicles that pinch off from the Golgi and are transported along cytoskeleton tracks. Some vesicles bind with the plasma membrane, secreting their contents outside the cell. Other vesicles, called lysosomes, contain digestive enzymes. Here, a lysosome fuses with a worn-out mitochondrion and breaks it down. Each of the trillions of cells in your body is a dynamo of activity, requiring millions of ATPs every minute. But most people are unaware of all this activity in their cells.
Do you think about all this activity in your cells?
I do.
Alright, I'm gonna show you a plant one, it's a little bit shorter.
Both animals and plants are made up of cells. Their cells have many features in common, but there are a few significant differences. Let's look inside a leaf to take a closer look at a plant cell. First, we encounter a protective cell wall outside the plasma membrane. The cell wall is made from strong cellulose fibrils. Once inside the plant cell, we see the large central vacuole, which regulates the composition of the cytoplasm, creates the internal pressure that is characteristic of plant cells, and stores various compounds produced by the cell. Plants make their own food by photosynthesis in chloroplasts. Light passes through the two membranes of the chloroplast and strikes these green discs, where light energy is converted to chemical energy. The sugar molecules produced by photosynthesis can be made into other molecules or broken down for energy. All plant cells have mitochondria, just like animal cells do. Sugars produced by photosynthesis are broken down and converted to ATP in mitochondria. Most organelles, like mitochondria, are found in both plant cells and animal cells. So the next time you pass by a plant, remember that we have more in common than meets the eye.
Remember that when we're not trying to do this. Alright, so, oh, they just kicked me off. I must mean that we ought to stop here. Alright, so we'll stop here and tomorrow we'll take a look at more of the organelles. I want to point out or talk about the packet you picked up here today. This is referring to what we talked about with surface area and volume. And so there are multiple problems in here about calculating surface area and volume, and it has different shaped quote-unquote cells, like cube-shaped and so on. So I put some formulas on here about how to figure out volume and surface area of different shapes, and so I want you to see if you can calculate it. So, so this packet is your homework. Okay?
What's the AP formula? Oh.